yours, John.
Okay, thank you, Piero, for inviting me. And I was late because I was at another function. I was trying to decide, do I go to that function or do I be late for this one? And I looked at the agenda and I whizzed through and I was like, oh, something about art, something about sustainability. And I was like, oh, I work in sustainability. That's good. And I was like, I think I can miss the beginning one on art. And then I walked in halfway through and looking at all these beautiful pictures and hearing about all this beautiful art, and particularly from the Muslim world where I've traveled quite a bit, in Iran where I've traveled as well. And I was like, this is why Piero brings together art and science, because you come and you find things that you don't always see and you don't always work on. So I really regret missing half the talk, and it's really good, so thank you for bringing these two worlds together. So I want to start by talking about what synthetic biology is and what SynBioBeta is. I work 50% of my time at NASA. I'm Director of Planetary Sustainability there, and we work on technology partnerships that make it cheaper and easier to be sustainable in space. And then we bring those technologies back to Earth and try and solve some of those sustainability problems that we have here around food, water, waste, and air. And the other 50% of my time I run SynBioBeta, and I do consulting with a number of startup companies. So it's an organization for synthetic biology startup companies, and I run events that bring together investors with startup companies, and we teach courses in synthetic biology, and we have a blog and a news digest. So I want to start by asking how many people here could tell me how a gene relates to a protein? And you can just stick your hand up if you could. And I'm not going to ask you to do it. If you stick your hand up, it's just going to help me to know how much detail to go into. So that's about 40% of people could say that they understand how a gene relates to a protein. So I'll just give a quick primer to in biology, and majority of this is for the other 60% of people. So cells are self-replicating machines. Inside of cells is DNA. The DNA makes proteins. Cells can replicate every 20 minutes. And one of my heroes, John von Neumann, the father of computer science, talked about self-replicating machines. Little did he know that we have actually self-replicating machines because the structure of DNA and the sequence of DNA was isolated. We have biological machines in every single one of your cells, and in every single one of these cyanobacteria or algae or bacteria. And biology is a manufacturing technology. In each cell you have multiple copies of DNA, and the DNA encodes for proteins these other functional molecules which actually do the things. They cut things, they form structures, they are colors that change with different wavelengths of light. So the proteins are the things that actually do the things, and the DNA is the stuff that stores the message. The cell is the chassis for the genome. The genome is the operating system for the cell. And I'm going to show you a quick video.
What you are about to see is DNA's most extraordinary secret. How a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene.
This is an animation.
A gene is simply a length of DNA instructions stretching away to the left.
But it's in real time.
The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
50 years ago, we didn't even know the structure of DNA. 10 years ago, we didn't know the sequence of the human genome. This technology, as I'm going to show you in a couple of charts, is increasing exponentially, both in our ability to read DNA, that is, you could go and sequence your whole genome for, I'm pretty sure you could do it for $1,000 these days. It was always the dream that we could do that. I'm pretty sure that we can do it for $1,000. But did you know we're going through a revolution in writing DNA as well? You could actually write out a complete chromosome, not of a human yet, but of a bacterial genome, and that was demonstrated by Craig Venter and his group about 2 or 3 years ago now. So synthetic biology is a bit like the web and Web 2.0, that you saw a lot more finesse done when Web 2.0 came along. Things seemed to be connected, a bit more logically connected together, and things seemed to work a bit better and look a bit more beautiful, and it got a lot of people excited and a lot more money into the field. And a bunch of people said, what is Web 2.0? It's nothing different. We've been doing it all along. So you got that similar thing with synthetic biology now. A lot of new blood into the field, a lot of new money from investors investing in the field, smarter ways of doing things, better ways, easier ways to engineer biology. And you've got the old guard saying, this is nothing new. We've been doing this for 30 years. But the core of synthetic biology is the process of trying to make biology easier to engineer and trying to turn biology into an engineering discipline. Just as computer science, if you look at the history of it, and you go back and you trace back computer science departments at universities, they invariably either came out of physics and mathematics, or they came out of electrical engineering. And if you trace back, you'll find the types of research being done now is a product of the history of where that department was spawned from. And it's the same with biology now. If you go to a genetic engineering department or a synthetic biology department or a computational biology department, you will get strands of those departments that are very much science-oriented and strands of those departments that are very much engineering-oriented. I'm more on the engineering side. I want to make things and I want to make it easier to engineer organisms. SynBioBeta is the company that I started. We had our first meeting in Menlo Park in 2012. We brought together a bunch of startup companies that nobody really knew existed yet. It's a new industry, and people wouldn't have thought there were enough companies to have a conference dedicated to synthetic biology companies. But we did, and it was very popular. We had a lot of these companies represented there, and we had another one just last November in Mission Bay, and we're running another one in London in April. So it's a hot field, a lot of investors in this field, and a lot of new companies. I'm going to talk about the types of companies that there are in synthetic biology and show just on this chart here how fast they're growing. There's an exponential increase in the number of synthetic biology companies. And you see them generally in these 4 different areas.
This is worldwide, United States, California?
This is worldwide, but the majority are in the Bay Area. And if you look at the total US bioeconomy now, and you look at just the amount of genetically modified stuff in the US bioeconomy, it accounts for more than 2% of gross domestic product. So, like it or not, in terms of genetic modification, it's contributing a significant amount to the economy in terms of crops, drugs, and chemical production. So I'm going to start with the bottom rung of this SynBio stack, which is gene and genome synthesis. This is a bit like the zeros and ones of a circuit board, but instead they're A's, C's, T's, and G's. you could type out a series of A's, T's, and G's on your computer, email them to a company, maybe 100 or 200 letters, and the next morning via FedEx you could have that DNA that's been printed out and holding it in your hand, ready to do your experiment. So there are a lot of companies in this field of gene and genome synthesis that is making DNA. It's the substrate for genetic engineering. These are oligos, is the name for small pieces of DNA, and there are different ways to assemble them and to make them. The market size is about $230 million worldwide. And it's pretty much an old technology. The chemistry is 30 years old and it hasn't changed that much. I showed a picture of Rob Carlson a second ago, and these are, this was coined by The Economist. They are Carlson's curve. Just like you've got Moore's law, the number of transistors on a computer chip doubling every 18 months. This is Carlson's curve, which shows that the cost of sequencing is halving every 18 months. And also the cost of synthesis, that is writing A's, T's, and G's, is also halving every 18 months. So you've got these kind of exponentials that are driving the field and making investors very excited about what the future holds. And here are just some of the companies. DNA 2.0 is down in Menlo Park. SGI DNA is in San Diego. GenScript is mostly in China. Genabler is in the UK. So next up on the rung is CAD tools for genome engineering. If you can write millions of bases of A's, C's, T's, and G's, what are you going to say with it? Well, at the moment we're kind of at the equivalent of the first word processor in terms of writing genetic code. So we were maybe using a basic script, and now we're at the word processor. There are some drag and drop interfaces for doing genetic design as well that people have come up with. So here are a number of companies that are working in the area of computer-aided design tools for gene and genome design. Next up we have organism engineering platforms. And Craig Venter, who I mentioned earlier, is one of the leaders in this field. His company Synthetic Genomics has a platform for doing digital to biological conversion. That is taking a sequence of A's, C's, T's, and G's and synthesizing a whole organism or the genome for a whole organism from that. And there are a number of other companies that have good platform technologies in this area. If you then look at the top and think about the applications, a lot of these are biofuel applications, that is conversion of carbon or some other CO2 or some other carbon product into a higher value fuel or a higher value chemical. And what you've seen is that a lot of these companies started from the green subsidies that were made available to try and shift us from a petrochemical fuel economy to a bio-based fuel economy. Once the subsidies ran out, a lot of these companies have shifted their infrastructure towards high specialty chemicals that you can sell for a lot more than you can fuel. So there's still a lot of work to be done in terms of making fuel cheap enough to replace petrochemicals. Biofuel's cheap enough. And this is outlined in a book that Rob Carlson wrote called Biology is Technology, which I thoroughly recommend. So who's winning in this game? Well, if you look at a lot of these IPO'd companies, they haven't particularly done very well. A lot of that's because of the volatile price of fuel, and they were in the biofuels market having to compete with that. So the stock prices don't look very good. Some of the people who are doing really well are the tool makers. It's a gold rush, and these people are selling the picks and the shovels. They're selling the pipettes and the tubes for you to do the experiments. So these market caps of billions of dollars for Thermo Fisher, 31 billion, and 12 billion for Life Technologies. So there's a lot of money to be made in the tools business. And then here are some of the companies that are funding some of these startups. You'll probably recognize some in the Bay Area. Founders Fund is just over in the Presidio. Google Ventures, Angel List, there's a fair amount of activity on synthetic biology. Khosla Ventures, a big clean tech fund. And it's a national initiative. The White House issued this report in 2012 trying to strengthen R&D in this area. And they see it as a new manufacturing initiative that just as we're losing out on traditional manufacturing to India and China, America could reinvent itself around bio-based manufacturing, taking in CO2 and converting it into high-value products. So I wanted to end and potentially start a discussion on GMOs. This was the cover of MIT Technology Review, by Fresh, by GMO. It's obviously designed to be provocative because the word GMO sends a visceral reaction through a lot of people. But it lays out a very, very nice case for GMOs. And I think the discussion that would be good to have is if you, how many people here take airborne if they're feeling sick? 1, 2, 3. So I was having this argument with 2 of my colleagues at NASA who are, we work in the area of sustainability, so we're always talking about climate change and climate change denial. And there's this whole slew of things, particularly at NASA. You know, I think half the country doesn't believe that we landed on the moon. Certainly half the country doesn't believe in evolution. I don't know what the current figure is for climate change. You've got some double-digit, I think, percentage of kids in Saratoga who are not vaccinated. And so the latest argument, and we have these rational science discussions, and sorry to those of you who take airborne, but if you look it up, the FDA sent a cease and desist letter to airborne because they said you have no evidence whatsoever of the efficacy of airborne. You have to take any words about health and it being good for you off your product because there's no evidence. So it might have been designed by a high school teacher, and that sounds great, but there's no evidence for it. So it's kind of the same for the now genetically modified foods. So while you might have a huge group who are proponents of the irrationality that the climate change deniers have for saying that climate change is a bunch of hooey is the same irrationality that the anti-GMOers have for saying that GMOs are dangerous. We've been growing GMOs now for over 20 years, multiple studies, multiple controls, and there's not a shred of evidence to show that there's any dangerous effect to the environment or to our health from taking them. However, when you look at the flip side of what GMOs can provide, they can provide crops that require far less fertilizers, far less pesticides, much reduced water, much increased tolerance to drought. So when you look at where we're heading, and this article talks about climate change will make it increasingly difficult to feed the world, it's very easy for us Westerners to say, no, go shop at the, don't grow your GM potatoes. I want to go shop at my farmer's market in Menlo Park. But try telling that to people who are starving and potatoes are their subsistence crop. And I don't think it's our right to tell Brazil or India, no, you can't have these products, you can't have these technologies because they're dangerous for you, when there's no evidence at all for it. We run a really good course, an introduction to synthetic biology. We've got about 4 of them planned in the Bay Area over the next few months. It's a 1-day course where we take people who put your hand up or who don't know how gene relates to a protein. We start at that level, and by the end of the day we have a very, very interesting discussion on GMO, which is a lot more balanced than the pro-GMO diatribe that I just gave you. But we have a very good discussion of that. And we've got a couple of new courses, Introduction to Carbon Central Metabolism and Big Data for Synthetic Biology. So thank you, and look forward to discussion.